In this video, we are going to look at the individual steps of the citric acid cycle. So if we go back to our metabolic pathways and we think about where we've been already, right? We have looked at glycolysis, where we looked at the conversion of glucose to pyruvate. We've looked at gluconeogenesis, which was the reverse of that. We have also come over here and looked at the fermentation route to get lactate from pyruvate. We have looked at the pyruvate dehydrogenation complex that gave us acetyl-CoA. And now in the citric acid cycle, we're going to look at how acetyl-CoA can shuttle energy into this cycle. So we're coming over here to look at this citric acid cycle right here. Okay, acetyl-CoA, and this isn't fully on here, but acetyl-CoA can also take energy to store it in fatty acids, but that's not today's topic. All right, let's take a look at this. So we're going to get oriented here. We are looking at the citric acid cycle, and this occurs in the mitochondrial matrix. And as we look, we're going to start with our acetyl-CoA, and that is carrying these 2 carbon units, that acetyl group, into the citric acid cycle. Okay, we can make that acetyl-CoA from glucose, so that glucose would go through glycolysis to pyruvate and then through the pyruvate dehydrogenation complex to form that acetyl-CoA. We've looked at that. All right, it can also be formed from fatty acids or amino acids, which we haven't really looked at yet. The citric acid cycle is going to convert energy from that 2 carbon unit in the acetyl-CoA. It's going to convert energy into, an ATP, into some high energy electrons that are going to be carried by NADH and FADH2 into the electron transport chain, and it's going to generate some carbon dioxide waste. All right, once these electrons move into the electron transport chain, which will be next week's topic, those electrons are going to be used to reduce this oxygen. Okay, and energy from that reduction process is going to be used to pump protons out of this matrix. Okay, so they're going to go through the inner mitochondrial membrane. They're going to be pumped out of that. And then that protein gradient that is generated by that will be used to power this ATP synthase to generate ATP. So the part on the right here, this electron transport chain part, that is next week's topic. What we're going to focus on today is how this works, which is what generates those electrons that we can send into that electron transport chain. All right, our first simplest look at the citric acid cycle. Okay, it has 2 stages. The first stage is 5 steps. The second stage is the last 3 steps. So there's 8 steps total. Here, we'll write that down. So we got 8 steps. So stage 1 is 5 steps. And then stage 2 is the last 3 steps. Okay, there's a lot of writing on this, so we're going to go through it one piece at a time. Okay, and then, so first with this picture right here, we're going to look at the big picture of the citric acid cycle. And then we're going to dive down and look in a little more detail, and then we'll finally get down to an individual step-by-step detail. All right, but let's look at this piece. Oh, and it did not highlight. That is so weird. It is highlighted in my slides. Let's highlight some stuff. So we want to look at this piece. So in the first stage of the citric acid cycle, 2 carbons are introduced, so that's happening right here. And they're reacting with our oxaloacetate to form citrate. So that's our first step right there. Okay, if we look at our second step then, that 6-carbon compound undergoes 2 oxidative decarboxylations. So let's take a look at that. So as we come through here, notice it is generating a carbon dioxide and again generating a carbon dioxide. Okay, so our molecules being oxidized, and we're going to look at that in detail, but we could tell it was being oxidized if you look at this. We're generating NADH, so the NAD plus is being reduced. It's gaining electrons. Our molecule must be losing electrons for that to happen. All right, so that gets us through the first stage. And then at this point, so we're going to start the second stage right in here. All right. And in that second stage, we're regenerating our oxaloacetate. So as we go from here to here, we're regenerating this piece we need to continue our cycle. Okay, both of these stages, let me pick a better color for this one. Both stages generate high energy electrons, and we can see that lots of places here. All right, lots more slides we don't need. I had them all, I thought, highlighted for you. Okay, so now let's look at this in more detail. Lots more detail, right? Let's see if we can make it a little bigger. First of all, when you hear citric acid cycle, know that there are other names for it. You may have heard it called the Krebs cycle, okay, after the person who discovered it. You may hear it called the TCA cycle, all right, TCA, tricarboxylic acid cycle. And if you want to think about why that is, look at our citrate molecule here. We've got carboxylic acid, carboxylic acid, carboxylic acid group. So tricarboxylic acid cycle. So all three of those you might hear. I think citric acid cycle, Krebs cycle are much more common. Okay, so now we have this more detailed picture. Let's take a look at what's happening. So in this first step, our citrate synthase, okay, there's our enzyme, is helping combine the acetyl group right here, okay, from the acetyl-CoA with the oxaloacetate. All right, so we're forming those. And so when you come over here, notice that we've got this one in green. There is this piece that came from that coenzyme A, the acetyl coenzyme A. So it gets tacked on there. So we have our original oxaloacetate molecule with this acetyl group, or this carboxylic acid group added right on here. All right, then in our next step, we have this aconitase enzyme, and it is moving the hydroxyl group. So take a look. Here our hydroxyl was on that carbon right there, and now it is moved up to this carbon. So there's the big change. And this is setting it up so it can better undergo oxidation in the next thing. So our next piece right here, dehydrogenase, you're going to think redox reaction. So here we have oxidative phosphorylation. phosphorylation. Okay, from that isocitrate dehydrogenase. Whoops, that is not what I wanted. Enzyme right here. Okay, we can see, again, NAD is being reduced. It's picking up those electrons to be NADH. So we knew it's being reduced. So we know that our compound must be being oxidized. And we can see that right here, right, where we're going from that hydroxyl group to the carbonyl group. So we can see that oxidation. Then so our product from that step is this alpha ketoglutarate. And if you think alpha keto, well, so when we look at our carbonyl group, our ketone group, right here, it's in the alpha position, alpha keto. All right, in step 4, we have our alpha ketoglutarate, and it is going to, our enzyme here, named after this step, right? Alpha ketoglutarate dehydrogenation complex. We're going to look at this in more detail in a little bit. We're going to see that it's very, very similar to the pyruvate dehydrogenase complex that we looked at in the last video. Okay, so in this step, notice that we again have NAD being reduced, so our compound is again undergoing oxidation. Okay, so in this time when it's oxidized, okay, we're spitting off this carbon dioxide and we're adding this thioester bond, this high energy thioester bond. So this process of decarboxylation gives off energy, and we use that energy and kind of trap it in this high and remember this is called a thioester linkage or a thioester bond. Right there. All right, we get to step 5. We have our succinyl-CoA synthase. And what we see happening now is that thioester bond is being broken, and we're going to use the energy from breaking that bond to form ATP. Okay, so this is substrate level phosphorylation to generate that ATP. So here is the end of step 1 or stage 1, right? So at this point, we have finished stage 1. And now, though, we need to regenerate our oxaloacetate so that we can continue the cycle. So when we look at the next step, okay, we have the succinate dehydrogenase. We're going to oxidize. So look at what we're doing. We had a single bond here, and now we have this double bond. Okay, we're oxidizing the carbon bond to form this double bond, and we're creating some more high energy electrons. We're going to look at that piece pretty closely in a little bit. Okay, step 7, fumarase. So at this point, we're going to add water across that double bond and to form the hydroxyl group. Okay, so we've added our water there. And then, as we get back to this last step, malate dehydrogenase, we are again creating some high energy electrons, and we are finally back to our starting material. All right, so now that we've gone through this series of steps, I have posted a video. It's an animation that shows the it will give you an appreciation for the speed and complexity, the density of the proteins in the mitochondrial matrix. So when you're looking at the video, and I'd recommend you pause, keep this screen up so you can think about our individual steps and watch the video. The big things shown will be the enzymes. Enzymes are big molecules, and you'll be able to see the small molecules moving in and out of the active sites. All right, so take a minute and do that. And then we are going to look through these steps one by one. All right, so step one. So first of all, let's just talk about the citric acid cycle in general. It is a very central pathway, very well understood enzymes and substrates, and so we can really look at each of these in turn. Step one is a two-step mechanism where the methyl group from the carbonyl carbon over here, or the methyl group is going to attack the carbonyl carbon. Whoops. Okay, when that happens, right, we're going to see that these electrons will swing out here. They'll capture the hydrogen. Okay, and so we get this intermediate citryl-CoA where we've bonded those two together. And then we're going to hydrolyze off that thioester bond. Okay, so we get our CoA back. Okay, we're essentially breaking it right there. The energy given off is going to drive the synthesis of this 6-carbon citrate molecule. Okay, so the breaking of that bond helps drive this process where we make this citrate. All right, step 2, iconitase isomerizes citrate to isocitrate. This is interesting because you would think that aconitase would be an isomerase. If you remember our classes of enzymes, isomerase. But even though that isomerization's the result, look at the steps that we're doing here. First, we're eliminating water, right, to form this double bond. And then we're adding water back in to go back to this piece. So it turns out it is actually a lyase. Okay, but you wouldn't know that if you didn't know the steps, if you didn't look at the steps. All right, step 3, isocitrate dehydrogenase catalyzes the oxidative decarboxylation. So think about that. It's being oxidized. We're decarboxylating. We're taking off that carboxylate group of isocitrate, forming alpha-ketoglutarate and capturing the high-energy electrons. All right, so the first step, it's again 2 steps. The first step is our oxidation. So look at that, right? We know NAD is being reduced, so something has to be oxidized. And look, here we go, right? We see that our carbon is oxidized. And then as we move over here to the right, in the second step, we're going to see that we are decarboxylating this. Okay? So we've produced high energy electrons, we've released carbon dioxide. This is an energetically favorable reaction. And we're going to move into step 4, where that alpha-ketoglutarate from the last, the product of our last reaction is converted to succinyl-CoA. All right, so this is another oxidative decarboxylation. So look at it. We are going to be taking we're decarboxylizing it, and we're going to harness the energy from that decarboxylation and trap it in this new thioester bond that we're making. Okay, so that bond is high energy. It is thermodynamically unstable, high energy, it can give off lots of energy, but it's kinetically stable. It can stay this way until we have the appropriate enzyme to help break it. All right, this alpha keto, let me find a good color to this right here. Alpha keto glutarate dehydrogenation complex is incredibly similar to the pyruvate dehydration complex that we looked at before. Whoops. Okay. It is so similar that enzyme 3 is exactly the same in both. And it has exactly the same function in both. Okay? It regenerates the FAD and it regenerates that lipoamide. Remember that one that served as a carrier to carry substrate from active site to active site, that swinging arm? Okay, enzyme 3 is the same in both of these. And we see we need the same cofactor. That coenzyme A is the same cofactor, just like in the pyruvate dehydrogenation complex. All right, step 5. We are more than halfway through here. Okay, this synthetase, usually when we see that, and where do I have it written here? Here we go. Usually when you see this type of enzyme, so the succinyl-CoA synthetase, it's usually a step where we use ATP to drive the reaction. In this case, we are actually driving the reaction in the direction to make ATP, to form ATP. But this is a reaction that can run both ways. So it'll run in reverse when the organism needs the succinyl-CoA. And you might wonder why in the world you would need that. It turns out that this is the starting material for heme. So your body has a little bit of a competitive cycle here, right? Depending if you need to keep going forward through your citric acid cycle and make more high energy electrons for our electron transport chain, you're going to want to drive this reaction this way. On the other hand, if your body's like, you know what, we really need to make more heme, I need this, and I've got a bunch of this, you're going to want to drive this reaction in reverse. So we're learning the pathways individually, but notice how complex and interrelated they are. All right, let's take a look at the mechanism for the succinyl-CoA synthetase. All right, so we have, just orient yourself here. The yellow part is your enzyme, and this histidine is an important piece of that active site. So it's part of our enzyme. So the phosphate can come into the active site and is kind of protected in this active site. And that phosphate is going to get used to cleave this high energy thioester bond. All right, so if you're looking, the phosphate is going to attack that carbon. So these electrons come up here. All right, and we see that our coenzyme A can now leave. Okay, and we've broken this high energy bond. So in the middle, we see this intermediate that now has the phosphate bound to that succinyl molecule. So we have succinyl phosphate. And we see that the nitrogen's going to come in here and attack and form a bond. So when it does that, right, so this attacks here, that causes that bond to break, And now we see that our succinate can leave. But we don't yet have our enzyme back, right? Now we have this histidine that has this phosphate piece bound to it. All right. And down here now, we see that an ADP will enter the active site, and this phosphate will get transferred to that ADP. That's where we make our ATP, and now we have our enzyme back. Okay, and this enzyme is reversible. So this enzyme can run in both directions, so we can make our succinyl-CoA, or we can make the succinate and the ATP. All right, once we're there, we're finished with stage 1, and now we are to stage 2, which is all about regenerating the oxaloacetate. Okay, when we do this, so I've got all 3 stages right here, step 6, 7, and 8. Okay, in step 6, we're oxidizing the molecule, so you can see this formation of this double bond. We can see that FAD is being reduced, right? So it's taking the electrons. We're going to come back and talk more about this. So this is this succinate dehydrogenase. We're going to come back and talk about that piece in just a minute. Then in the next step, fumarase is our enzyme. Okay, and we're essentially adding that water, and this will look very similar to ones that we've done before, right? To make malate, which you have seen before in other pathways. And then in step 8, okay, we're making some more high-energy electrons. All right, we're oxidizing that carbon. This is another reversible step, this malate dehydrogenase. Right now we're looking at it in the citric acid cycle forward. But if we remember back to gluconeogenesis, and we talked about pyruvate and how oxaloacetate would form, but it needs to be shuttled out of the mitochondrial matrix. And if you go back and you look, you see what happens is that this runs in reverse. Oxaloacetate forms malate. That can make it out of the matrix, and then it got converted back to the oxaloacetate so that we could continue our gluconeogenesis. All right, this has been a lot. We are going to look right here at just a couple other things about the citric acid cycle. So this is another picture of the citric acid cycle. Similar, just a different diagram that highlights a few different things I want to point out. First of all, in this middle part, we have highlighted the the carbons that came from the acetyl-CoA. So essentially, these carbons. And those carbons are shown in the red. And notice here they are, here they are, here they are, here they are. And the key thing I'm just pointing out here is that these 2 carbons are not spit off as carbon dioxide. Remember, some of our carbons are leaving as carbon dioxide in these various places, there and there, okay? But it is not in this first round those 2 carbons that were just added. And they will be spit off in the next cycle or 2, depending on the orientation of the molecule. So they're going to make it all the way around the cycle, become a part of the new oxaloacetate molecule, and then in the next cycle or 2, they will be spit off in those carbon dioxides. All right, that was one point I wanted to make. Another point, this looks a little different because notice we've got GTP. And when we looked at it, we talked about ATP being formed, and that can cause confusion. But just know that some cells will form GTP, So if they're running, say, a route to get biosynthetic energy versus just energy to keep the cell running, different types of cells have different versions of the succinyl-CoA. So liver cells tend to have a version that makes GTP, while muscle cells tend to form ATP. So different versions of this in different cells for different purposes will make GTP or ATP. Simpler organisms tend to do one or the other, not both. Oh, I forgot to go back to this. So we're going to look at this. Look right here. This is interesting because when we looked at the earlier one, it had FAD to FADH2. Let's go back up and look at this. So the FAD picks up those high energy electrons, and it needs to shuttle them to the electron transport chain, but the FAD is bound as a prosthetic group. So remember that term? Bound as prosthetic group. So it is stuck on this enzyme. So it can't just swim over to deliver the electrons. So instead, those high energy electrons, so here's that complex, okay, of the enzyme with the FADH2 now bound. It's going to transfer those electrons. Q here, all right, stands for ubiquinone. Okay, and when it picks it up, ubiquinol. So this molecule now can move over and deliver those electrons. It is not water soluble, so this ubiquinone and ubiquinol exist in the inner membrane bilayer. So this reaction is all happening in the mitochondrial matrix, and that matrix has an inner membrane, and these non-water-soluble groups can exist in that middle of that membrane, that bilayer, and they can diffuse around in there. Okay, this enzyme is conveniently also attached to the inner matrix membrane. All the other enzymes we've looked at are inside the matrix floating around, but this one is stuck in that membrane. And you'll see that in that video we looked at. There's a picture of that. All right, so that was a very big overview of the citric acid cycle. So next up, we'll look at regulation and how the material needs of the cell are met with citric acid cycle metabolites.